everyone. I am Dr. Sara Kashkouli, and welcome back to our How to Build a Human Embryology series. Today is our second lecture together, looking specifically at the development of the respiratory system. So as before, I'll share a general sort of roadmap of the sort of steps, the things we're going to look at during our lecture today. So of course, we'll start with a little bit of a quick recap of our first lecture on embryogenesis, just in case you might have forgotten some of the key elements there, to kind of start to provide a little bit more context to the development of the respiratory system. And then following this, we'll dive right into the development of the respiratory system itself. Now, this can be divided into 5 main stages, and so we'll go through each of those stages individually, and then we'll kind of go a little bit deeper by discussing circulation and kind of the connection that exists between the respiratory system and the circulatory systems. Now, we haven't done, of course, embryology of the cardiovascular system yet, but this sets us up a little bit to start to be able to answer some of those questions when we do actually get into the development of the cardiovascular system. And finally, we'll just conclude the lecture by talking about the important role the diaphragm has during breathing. So some of the objectives of today's lecture are that by the end of it, you should firstly be able to understand the development of the respiratory system and be able to explain each of those stages of its development. You should also be able to describe the role of surfactant in respiratory function. And lastly, you should be able to identify and start to generally understand some common respiratory congenital disorders that might exist. Okay, so we'll begin right off the bat with our recap. So if you remember during those early stages of development, right, when we had that bilaminar disc, so the epiblast and hypoblast layer of cells, these cells, well, technically, if you actually go all the way before that, even in the earlier stages of development, you had 2 distinct cell masses, right? You had an outer cell mass and you had an inner cell mass. And then we said if you just look at the inner cell mass, those cells start to reposition themselves to give rise to what is known as the bilaminar disc or the double layered disc. That's what we're looking at in this image here. So the bilaminar disc is composed of the epiblast layer of cells and the hypoblast layer of cells. Now, the significance here of the bilaminar disc is that it's going to undergo a very important process that occurs in the 3rd week of development, and this is known as gastrulation. Now, if you remember what gastrulation is that during gastrulation, this bilaminar disc starts to differentiate to establish 3 primary germ layers. So it becomes a trilaminar disc, right? Those 3 layers. And those 3 layers are, of course, the endoderm germ layer, the mesoderm germ layer, and the ectoderm germ layer. And that is the main event of the 3rd week of development. Now, as a result of a whole lot of very, very detailed, intricate cell signaling events, what happens is that a sort of thickening starts to appear on the epiblast layer of cells, and eventually it's this thickening that allows the migration of the epiblast layer of cells to kind of go in towards the hypoblast layer of cells. So not really getting into too much of the nitty-gritty of the apoptosis that occurs here and the cellular events that result in creating that sort of deepening in this general region for those epiblast layer of cells to start to migrate. But basically, just kind of remembering that these epiblast layer of cells, as a result of this groove that appears here on the primitive streak, they're now able to basically have a pathway to immigrate or move down or migrate and reposition themselves, and then eventually be able to kind of also kind of go through that or journey of differentiation, right? So this epiblast layer of cells here in blue is going to start to migrate down the primitive groove to make their way through all the way to the hypoblast layer, so this yellow layer of cells. So when they reach this hypoblast layer of cells, these newly migrated epiblast cells are going to start to displace first and then replace the hypoblast layer of cells. And in effect, by doing this, it's them kind of forming a new layer of cells. So as these epiblast cells kind of migrated downwards, displaced and replaced that hypoblast layer of cells, they start to form a new layer of cells, right? So they adopt a new identity. And because these epiblast cells, they're technically no longer epiblast cells, right? Because they migrated down, they made a home somewhere new, they displaced and replaced the hypoblast layer of cells. So they now kind of are known as the endoderm layer of cells over here. And so because you have this brand new kind of layer of endoderm cells, whatever initial epiblast cells that kind of migrate downwards and start to replace and displace and replace the hypoblast, those become the endoderm germ layer of cells. And when this endoderm germ layer is fully, completely formed, what's going to happen is that whatever remaining epiblast cells want to also start to kind of migrate downwards. Excuse me. But when they migrate downwards, they realize that, oh, they can no longer, become endoderm cells, because this endoderm layer eventually gets fully formed and doesn't accept any more new epiblast cells. So instead, these newly migrated epiblast cells, they essentially also start to settle down and create their own new territory, their own new layer of cells, and they call themselves the mesodermal cells, or the mesoderm cells. And this is how you have the second layer, the second of that primary germ layers, right? So you had endoderm first, and now we have mesoderm. But we can't forget what remains of the epiblast cells all the way at the top. Given that all the cells that wanted to leave left, and they created the endoderm germ layer, they created the mesoderm germ layer, so anyone that wanted to go went. Now, whatever you have left at that top layer of what used to be a full epiblast layer, this essentially undergoes its own transformation, or its own rebranding, if you will, and gets to be now the ectoderm germ layer of cells. And that really is like the super summarized version of describing how you go from a bilaminar disc with just that hypoblast and epiblast layer of cells to a trilaminar disc that is composed of the endoderm, the mesoderm, and the ectoderm germ layers. And that's essentially where we left off in our first lecture, kind of the establishment of these primary germ layers. And so with that, we can talk about we can start to talk about the development of the respiratory system and how the different aspects or the different parts of the respiratory system come to be. Now, with this, of course, supplemented by all your gross anatomy lectures that you're going to be having, as well as your anatomy hall, when you go into the dissection hall and you start to see the different organs that make up this system, you'll be able to put all these puzzle pieces together and have a clearer, larger picture of the whole of the respiratory system. And then as we go on, you'll learn that the respiratory system has two main parts, right? Its first fundamental component is its conducting portion, and it also has a second fundamental component known as the respiratory portion. So the conducting portion of the respiratory system conducts the air into the system, right? So it's going to be your first step in. So it's going to help to warm the air. It's going to help to moisten the air as it enters your body when you inhale. Now, the respiratory portion of the respiratory system, that's the important part of where that gas exchange is actually happening within the body. And of course, within this system is quite it stretches quite an expanse, right? So it includes the nasal cavity, it includes the oral cavity, it includes the trachea, the bronchi, the bronchioles, the alveoli, and each of these have their own sort of unique role in the entirety of this respiratory system and the whole process of respiration. And so we'll start to explore some of these structures in a little bit more detail. And of course, like I said, you'll add to your breadth of knowledge when you do the gross anatomy lectures, and then again later on when you do the cardiovascular system, the development of that, for example, you'll see how the respiratory system is connected to the cardiovascular system. And with each of these, as we go along, which each of these blocks you get to put together, you ultimately have one final image by the end of it. Okay, to basically simplify the entire journey through the development of the respiratory system, we can divide it into these 5 distinct stages. So you have the embryonic stage, the pseudoglandular stage, the canalicular stage, the saccular stage, and the alveolar stage. Now, from the top, the embryonic stage right at the beginning, right? So it's going to span those first 5 weeks of development, and that's what serves as our starting point here. Now, it's during this period of this embryonic period that we're starting to see a lot of the initial budding, the initial branching that's happening within the respiratory system. Next, we have the pseudoglandular phase, and this kind of is at this point where, we first in your embryonic stage, you started to see that budding, the branching, where by the time you get to the pseudoglandular stage, you start to see the culmination of that budding and branching. And so it's essentially quite an important phase in the system's maturation process. Then by the time you get to the canalicular stage, that's when you start to form the terminal bronchioles, which again, also significant in the development, the whole process of the development of the respiratory system. Now, the saccular stage is basically characterized by the development of these terminal sacs, as well as the establishment of some of the capillaries within the respiratory system. And then finally, you have the alveolar stage, which is happening all the way at the end. So about 8 months in utero, the alveolar stage begins, and it extends the development of it extends through to childhood. And it represents the stage where the blood and air barrier continues to mature until it reaches its, its final form and solidifies the functionality of the respiratory system. Now, of course, I've given kind of the weeks in which each of these stages occur, but for all intents and purposes, I don't really expect for you to memorize the nitty-gritty of when each stage is kind of occurring in, but rather to have an understanding of the principles of course, embryonic stage is going to be during the embryonic period of development, for example. What follows that is pseudoglandular. And as we later on in the lecture, we talk about things like surfactant and its formation, then it'll be kind of important to understand at what stage that is occurring and how that kind of explains kind of lends itself to explaining into how the entirety of the respiratory system works. So again, not to memorize the nitty-gritty of, oh, this is happening on X day or X week, but rather to give you the broad general principles of when each of these significant things are occurring during the development of the respiratory system. So we'll start right off the bat with the embryonic stage. Okay, so to give you a sort of general overview of the development process that we're going to be discussing, we'll take a closer look at this image. So this is kind of depicting a developing embryo. And at this stage, of course, we've only done the embryogenesis lecture, so a lot of this is completely new to you. But I kind of want to introduce it to you at this stage, and we'll keep coming back to this image in later lectures. But as a starting point, what I want you to notice is that the respiratory system over here in blue is actually derived from the developing what's known as the primitive gut tube. So this bit in yellow extending onto pink and then extending onto blue here at the end, this entire thing that spans the whole length of the developing embryo, this is the developing gut tube, so giving rise to the gastrointestinal system. And we come back to this, of course, when we do the embryology of that particular system. But at this stage, I want to briefly start to introduce to you that whilst this gut tube kind of develops as a sort of tube that runs the entire course of the embryo, it's also divided into little sections, which explains the different colors, so the yellow, pink, and blue. So what you have here are right at the top, you have the development of the foregut. In pink here, you have the development of the midgut. And in blue here, you have the development of the hindgut. And that's all you really have to know at this stage about this. But what's important here is if you look at this tiny little speck here in blue, that's the beginnings of the lung bud. Or the respiratory diverticulum. And as you can see, it's arising as this sort of outpouching of this region of the foregut here. And it's in this in these early stages of development, this diverticulum or this outpouching is going to maintain this sort of continuity that it has with the developing foregut. And eventually, as development progresses, in order for there to be functional stability, a separation starts to exist between obviously what will be the respiratory system and what will be the digestive system. And this becomes very apparent as we kind of progress through development. So in describing that separation between the respiratory system and the digestive system, this image here starts to describe that a little bit. Now, if we visualize just the topmost part of the foregut, from which we said the respiratory diverticulum or the lung bud starts to emerge, right? This is essentially what you're looking at. So around 4 weeks of development, the respiratory diverticulum starts to undergo a series of like sort of divisions or transformations to start to give rise to its various components within the respiratory system. So of course this would include things like the bronchi, the lobar bronchi, the tertiary bronchi, the bronchioles, and ultimately the alveoli. So all of this is starting to happen around week 4. But again, going back to the start, you have that continuity between the foregut, which the respiratory diverticulum or the lung bud juts out from or out pockets from. And so you need that separation to be established, right? So the separation is established by the formation of what is known as a tracheoesophageal septum. So this is quite an important step that occurs before any of these further divisions to the respiratory diverticulum can even take place, to start to create that separation between what will become the trachea and what will become the esophagus. So that's essentially what the name suggests, right? Tracheoesophageal, so tracheo from trachea, esophageal from esophagus, and septum meaning that barrier or that separation between the two. Now, again, it's worth noting that the surrounding region that's kind of that's enveloping this developing respiratory diverticulum is made up of mesoderm, right? And it's this surrounding mesodermal cells that play quite an important role in the development of important structures in this area, like the cartilage in this area, or the smooth muscle in this area, or the connective tissue within the respiratory tract. And this really starts to kind of highlight to you the sort of kind of intricate interplay that exists between the different embryonic layers, between those different 3 embryonic germ layers that we spoke about in the process of the respiratory system in particular. So let's zoom into this. Now, if we look at that a little bit more detail, so this is what we were looking at, right? The development of the tracheoesophageal septum. Now, here you have that little this thing that's jutting out here. This represents the outpouching of the respiratory bud from that developing foregut in the back. Now, this is giving you a frontal view into this, right? So kind of looking at it through this way. But then if we start to look at it, so these images, 1, 2, and 3, is if you're looking at it from a top-down perspective, this is really what you'd observe. So the schematic outlines all of those structures. So in those early stages of development, the trachea and the esophagus are just this one big continuous structure. And to be able to facilitate this sort of separation of these two important structures, these things start to appear first of all, right? So these tracheoesophageal folds, as they are known, folds, they start to emerge, and they start to kind of first they establish this sort of initial continuity, but like I said, this continuity needs to close off, and there needs to be separation of these two structures for proper development to occur, right? So first you have the folds. These folds quite quickly realize that they need to start to fuse together. So they get closer and closer and closer together to where, if you kind of, you look at this transition from image 1 to image 2, where you had just two separate tracheoesophageal folds before, now you have this fused tracheoesophageal fold in the middle. So they both kind of grow towards the middle so they can fuse with each other to form what is known as the tracheoesophageal septum. And this essentially, this significant kind of going from just folds that move in towards the midline and fuse together, this is what gives rise to that tracheoesophageal septum, which is effectively what is separating the trachea from the esophagus, then placing the trachea anterior to or in front of the esophagus, which again, of course, is an important configuration if you think back to the respiratory and the digestive systems. Now, this will again make a lot more sense as you start to learn your gross anatomy aspect of all of this. And then you have another layer of understanding that will come in when you start to observe all these structures on your silent mentors in the anatomy hall, where you'll see the respiratory system kind of at these early stages, at least, is sitting anterior to the digestive system. Now, in the event that this separation between the trachea and the esophagus, either if it doesn't completely occur, so you don't have this full separation, or perhaps this separation only occurs partially, it will leave a point behind where there is going to be continuity between the trachea and the esophagus. And this is the first clinical thing we'll discuss today, where we have the formation of a tracheoesophageal fistula. And then that just really means that if you don't have this full separation of the esophagus from the trachea, whether it's there's partial separation, if there's some continuity between these two structures, it's of course going to cause problems in life for that infant, because, for example, if you go to swallow food, there is a fistula, so that so then that food kind of these two systems, or the contents of them, start to mix with each other, which is not compatible. Now, so we started from the foregut. We had that outpouching of the respiratory diverticulum. Now we're in this third image here. So as development is progressing, we're reaching this stage where the bronchial buds can now develop. So we'll zoom into this part now. So as this sort of process of development unfolds further and further, the respiratory diverticulum, where you already have that separation, it's going to undergo sort of a bifurcation. And when it bifurcates, it's going to result in two distinct buds. So this is what you see before it gets to this developed stage over here. It's still small up here in this image to the top left. So these bronchial buds, this is what represents the very initial stages of what will eventually become the right and left primary bronchi, which of course are the foundational components of the respiratory system. And then when these primary bronchi are good and well and established, they will then undergo their own subsequent series of proliferative divisions to give rise to the secondary bronchi, and then eventually the secondary bronchi branch out even more to give rise to the tertiary bronchi, which then branch out even further to establish or create the bronchioles and the progressively sort of intricate network of respiratory structures. Now, this very intricate branching process starts to set the stage for the highly specialized thing that is the respiratory system so that it can serve its postnatal function. So by the time you get to this stage, we're roughly around week 5 of development, and here we get to the kind of this this budding or the tracheal bud as it's developing here has now split into two to give rise to the two bronchial buds or the primary bronchi you see here. So what happens next is, as I said, progressively kind of as development progresses or advances or goes on, these primary bronchi will undergo further branching to give rise to the secondary bronchi. And during this stage of sort of early development that we're still in, you start to see a sort of interesting asymmetry become more and more apparent in the structural development of the lung. So specifically in the right, the right bronchial bud, what's going to happen to it is going to give rise to 3 secondary bronchi, whilst on the left side, it's only going to give rise, or the left bronchial bud will only give rise to 2 secondary bronchi. So on the right, you have 3. On the left, you have 2 secondary bronchi. And these secondary bronchi, another way to name them are lobar bronchi, because they, as you can see in this image here to the right, the lung is made up of different lobes, right? So you've got 3 lobes on the right, you've got 2 lobes on the left. These lobes are divided, they're separated by these fissures. So 2 fissures on the right lung, 1 on the left lung. And again, this is a fact that you will learn further when you do your gross anatomy. And so it makes sense then that that primary bronchi will split into 3 secondary bronchi or 3 lobar bronchi to go to each of these lobes on the right, and then to 2 lobar bronchi to go to each of these lobes on the left. Now, so that is basically kind of your, by the 5th week, how you're going from the primary bronchi towards the secondary bronchi of the lungs. Now, after that, the secondary bronchi are going to continue that process of branching, and they're going to basically give rise to the tertiary bronchi. So this is if we kind of zoom in on that and see how or why that's needing to happen. So the tertiary bronchi, another name for them is segmental bronchi, because they have a quite an important role in kind of supplying the segments. So the segmental bronchi are going to supply all of these individual bronchopulmonary segments within each of these lungs. Now, these bronchopulmonary segments represent functional divisions of the lungs as well as anatomical divisions of the lungs. And each of these segments really, for all intents and purposes, is quite capable of functioning independently and contributing to the efficacy of the whole entirety of the respiratory system. And so how you go from primary bronchi to the secondary bronchi or the lobar bronchi to then the tertiary bronchi or the segmental bronchi, ultimately, by the time you get to the tertiary bronchi, that marks rather the sort of the end of the embryonic stage of the development of the respiratory system. So the next stage we start to go into is the pseudoglandular stage. Now, by the time we get to the pseudoglandular stage of the development of the respiratory system, the respiratory tree itself, as it's known, because again, it's a lot of branching that's happening, that's going to undergo quite a bit of extensive division or branching, and ultimately give rise to further sort of 16 or 20 generations of bronchial structures by the time we get to the end of this pseudoglandular stage. So if we start from the tertiary bronchial, the tertiary bronchi, because that's where we stopped at the embryonic stage, right? So starting from the tertiary bronchi, you see the emergence of these smaller and smaller and smaller and smaller and smaller bronchial buds that start to branch off and they start to proliferate, that eventually leads to the formation of the bronchioles, which in turn become the terminal bronchioles. And that's what you can see in this illustration here as well. So you had the main bronchus that becomes a primary, secondary, tertiary. So primary, secondary, tertiary, which are the segmental bronchi here. Those then become the bronchioles, which then branch off into these tiny, tiny terminal bronchioles. So the tertiary bronchi essentially serve as the origin point for this sort of branching process that continues up until the point of the terminal bronchioles and until the terminal bronchioles are fully formed. Now, at the same time, within each of these bronchopulmonary segments, the ducts within these segments also start to develop. And it's this sort of ducting that exists, this piping that exists within the substance of the lungs that serves as that passageway that represents the sort of initial appearance of the respiratory elements. And this emerging passageway is of course characterized by the presence of a very specific type of respiratory epithelium that's known as ciliated columnar epithelium and simple cuboidal epithelium. And these feature these little cilia, C-I-L-I-A, that play quite a significant role in the respiratory process. Again, not to go into too much detail on this because you will learn a little bit more about this when you do the histology aspect in your gross anatomy classes. Now, as development progresses, it will eventually involve the gradual appearance of cartilage and smooth muscle within the bronchial walls. You also start to see the emergence of very lung-specific type 2 alveolar cells that are going to be responsible for the production of surfactant. And surfactant is a very important substance that's essential for the proper functioning of the lungs. And it's kind of the culmination of all these developmental changes that is reflected in the histological appearance of the lung as well, which as I shared, you will discuss a little bit more about in your gross anatomy classes, that very much gives the lungs a sort of gland-like quality. And that's why this phase is referred to quite commonly as the pseudoglandular stage. And so by the time you get to the end of this pseudoglandular stage, this sort of intricate branching and structural formation of the respiratory system will start to essentially reach its completion and set us up for the further sort of maturation and the functional maturation and specialization of the lungs itself. So the next stage is going to be the canalicular stage of development, where in this stage of development, we start to witness the specialized development of the respiratory components of the lungs. And so a couple of significant things start to occur here. So we have those terminal bronchioles, right? And then from there, we see this sort of further branching that occurs from these terminal bronchioles into these respiratory bronchioles. And again, you had those ducts that were branching out from the bronchioles as well, right? And these newly formed alveolar sort of ductal system that you see here, that is what ultimately feeds into the ends of each of these alveolar sacs that you see. And that's what's going to be responsible for the exchange of gases that's going to occur within this respiratory system. And so with the development of the respiratory bronchioles at this stage, you also slowly start to see kind of an increase in vascularization around each of the region of these respiratory bronchioles, which basically, so you have more, or you have an influx of pulmonary capillaries that come in to allow for that super efficient gas exchange or gaseous exchange that's going to happen. And quite notably during this stage as well, these canals and this ductal system that's forming, like the ones in the bronchi and the bronchioles, these also start to widen a little bit so that they can start to slowly adapt to their function. And that's why this stage in particular is called the canalicular stage. So it's starting to kind of focus on the formation and the maturation of the canals that exist within the respiratory system. And then as we kind of start to go on with this stage, we see that these canals or these tubes, they start to branch out a little bit more, and they start to kind of, as they emanate out from these terminal bronchioles, we start to see that they start to kind of, they constitute the formation of these specialized group of structures or units known as the acini, singular being acinus. And each of these comprises of a terminal bronchiole, an alveolar ductal system, as well as a terminal sac. And all of this, as it comes together, essentially can be described as the primitive early stage alveolus. And so this primitive alveolus basically represents kind of the premature precursor, or rather the immature kind of precursor to the final alveoli that we have, that is going to be necessary for gaseous exchange to occur. So, and like I shared in the previous slide as well, the ducts or the lumen of these ducts are going to kind of progressively get wider and wider. The epithelial cells within these sort of primitive alveoli, they're going to undergo transformation as well. Gradually, they start to flatten out to assume the characteristic of what's known as type 1 alveolar cells. And it's these type 1 alveolar cells that are going to help to facilitate the gaseous exchange that's occurring within the respiratory system. We also have those pulmonaries come in to kind of invade the region that surrounds each of these primitive alveoli. And so that by the time you start to inch towards the end of this canalicular stage, some of the primitive alveoli at this point could have potentially developed to a point where it's sufficient, where they're sufficiently developed enough, and they have become vascularized enough as well to begin to start to support some type of gaseous exchange. And this is quite it's considered quite a developmental milestone, and it's significant because it signifies that even though we're still kind of early in development at this point, if a fetus is prematurely born at this stage, at this early stage of development, it could still very much have a chance of survival, of course, with sort of very intensive medical intervention, but it could have a chance of survival nevertheless. And then next we move on to the saccular stage, which starts around 25 weeks into development all the way through to birth. And this is when the terminal sacs and the capillaries really come in and really start to develop fully and get ready for their functionality. So in this saccular stage, these alveolar ducts are going to give rise to these alveolar sacs. And it's these alveolar sacs that are very much instrumental in kind of essentially kind of finishing out or completing the structure of the respiratory system. So it's also during this stage, actually the saccular stage, that these primitive alveolar sacs continue to increase in number as well. And they start to develop a lot more extensively. So within these alveolar sacs, we spoke about this a little bit just now, but within these alveolar sacs, we have these type 1 alveolar cells, which, as we just said, they're essential for helping to facilitate that gas exchange, and they are simple squamous epithelia in nature, and they form a sort of vital component of the blood air barrier that's going to develop a little bit further at this stage. And then this barrier represents the sort of interface that exists between the epithelial type 1 alveolar cells and the endothelial cells of the capillaries to allow the facilitation of that gaseous exchange. Of course, that kind of simultaneously at the same time, the available surface area for this gaseous exchange to happen is also increasing, right? And that's also going to enhance the functionality of this system. Now, another significant development that's occurring is the development of the type 2 alveolar cells, which, as we mentioned a little earlier, is that these cells are going to take on the role of producing surfactant. And these type 2 alveolar cells are basically these simple cuboidal cells, and the surfactant that they produce is very important because it helps to reduce the surface tension of the fluid within the lungs, right? And by doing so, it's the surfactant that helps to prevent the collapse of the lungs or the caving in of the alveoli, and to ensure that the respiratory system maintains its efficiency and functionality. So we went from having these sort of simple cuboidal cells that line the primitive alveoli to now having two different types of cells, right? The type 1 and the type 2 alveolar cells. So the type 1 alveolar cells, as you can see in this image here following this legend, they're flat because they're needed for gas exchange, that gaseous exchange to occur, so that the blood cells on the the blood vessels can start to basically line the outside of these cells to allow for that efficient gaseous exchange to occur. And then these sort of cuboidal type 2 cells that we have, these are a bit more specialized, and these produce surfactant, so that when air kind of enters the alveoli and the air and fluid interface creates that surface tension that's going to cause the smaller alveoli to collapse. The surfactant is this phospholipid layer that's going to line the inside of each of these, each alveolus, to help to reduce that surface tension by binding to the water and the air within the alveoli to prevent it from actually collapsing in on itself. And then finally, we have the alveolar stage of development, which starts just before birth, around about the 8th month of development in utero, and it continues all the way through to childhood, or roughly about 8 years of age. So in the previous stages of development, we essentially started to establish the kind of groundwork with looking at the formation of the primitive alveoli, right? But for efficient respiratory function to take place, these alveoli actually need to mature. And they don't just need to mature, they need to increase in number, right? So the demands of the respiratory system require from it that for there to be many, many, many alveoli, so that by the time of birth, there are millions of alveoli present, and this maturation and this proliferation of the alveoli is primarily what occurs during the alveolar stage. And then kind of as the respiratory system is continuing to develop as well, you also have a continuous increase in kind of, as you can see in this illustration here, you have an increase of these pulmonary capillaries that play an important role in facilitating that gaseous exchange within the alveoli, to ensure that this sort of rapid maturation and the proliferation of the alveoli is also adequately supported in their function. So this alveolar stage is quite important in ensuring kind of that the respiratory system's its function and its structure starts to mature to meet this kind of sort of expanding, growing demand of this developing baby. And so that really brings us to the end of the development of the respiratory system. So we'll briefly start to put all those ideas together and start to summarize what we've just discussed. So we started with some bronchial branching that occurred early on in development. So that sort of branching is occurring here. Then we have our pseudoglandular stage of development, where the branching starts to finish off so that we can, the lung starts to look sort of like a gland. That's why it's a pseudoglandular stage, so that we can then proceed to the canalicular stage, wherein the terminal bronchioles start to develop buds, and branching furthers even more, so that you can start to transition into the saccular stage, where the terminal sacs appear, the capillaries appear, and then you finally get to the alveolar stage, which basically is, you sort of have the development, or the finishing. You have development finishing rather, and millions and millions and millions more alveoli starting to appear to facilitate that efficient sort of gaseous exchange. So what happens next? So after all of this is complete, we need to have circulation, right, for gaseous exchange to take place. So we'll look at that next. So during fetal life, the lungs are at that point filled with amniotic fluid, and the alveoli, the tiny air sacs that are responsible for gaseous exchange, they are in this sort of collapsed state. And it basically remains this way, kind of, it persists while the developmental process starts to unfold a little bit more in different parts of the body and the different organ systems of the body. So if we start to think a little bit about the development of the heart, again, I know we haven't done the embryology of the cardiovascular system, but these two are kind of related to each other, and we'll revisit this, of course, again as we do the development of the cardiovascular system. So the fetal heart itself, as it's developing, it has a couple of different shunts, all right? It has a couple of different shunting mechanisms that help kind of play a significant role in the blood that's circulating within the fetus from bypassing the lungs. So the fetus, as it's developing, it's primarily receiving its oxygenation from the placenta, right? Which is the organ that grows, that supports it, that gives it its oxygen, removes its carbon dioxide, gives it nutrients, removes its waste. That's what the placenta is doing. And it's also primarily what is helping to oxygenate the fetus. So again, we'll talk about these shunts that exist in the heart when we do the cardiovascular development. But then during this development, like I said, everything is kind of bypassing the lungs. The lungs are not functional at this stage. They remain collapsed, right? The alveoli. But a sort of remarkable transition occurs at the moment of birth, where when the baby goes to take its first breath, the amniotic fluid within the lungs, it gets resorbed by the pulmonary capillaries. And when this happens, it leads to a very notable drop in pulmonary vascular resistance, which in turn causes those shunting systems of the heart that help the blood to bypass the lungs, right? This drop in pulmonary vascular resistance causes the shunts of the heart to snap close. And when all of these happen, as well as the fact that these previously collapsed alveoli now have surfactant. So all of this is happening at the same time, so that when the baby takes its first breath of air, the lungs are going to be able to fill up with air, and the blood can start to efficiently pass through the lungs to enable kind of that essential process of gaseous exchange to occur. And this quite transformative event really marks the beginning of independent respiration for the newborn baby. Okay, so we've spoken about respiration, and of course we have to talk about the main muscle that's helping with respiration, and that is the diaphragm, and what it's doing and how it develops. So one of the components of the diaphragm is the septum transversum, which you can see here in this orangey color in this image. Now, around quite early on in development, around I think about 3 weeks, about 22 days into development, septum transversum first starts to appear. So this is a developing embryo. Everything is super primitive at this stage. You have the developing spinal cord, the developing vertebral body. None of them have the shapes you're familiar with. They're kind of tubes within tubes. You have the esophageal mesoderm developing, the blood vessels developing, the foregut developing. So everything is quite primitive at this stage, right? And that's when septum transversum starts to develop, and it's kind of in the region of the cervical vertebrae sitting caudal to the developing heart. And this initial position that it takes is quite important in developing or in determining the diaphragm's eventual innervation. Again, you're going to learn all of this further in your gross anatomy lectures, and it's going to start to make a lot more sense. So as development progresses, as the body starts to, because again, now it's just sort of like it's flat and all the things within it are tubes, but the body is going to undergo folding, right? We spoke about the folding of the body, the bending of the body. As that progresses, the diaphragm, even though it starts to develop in kind of the region of the cervical vertebrae, it's eventually going to shift downward to its ultimate location, which aligns more with the region of the thoracic vertebrae. And at the same time, the septum transversum is going to emerge as essentially the defining boundary between the thoracic cavity and the abdominal cavity, and contribute to the structural integrity of the diaphragm. And so we just said that the diaphragm starts to develop in sort of the region of the cervical vertebrae, right? So it's because of its initial development at that higher position that it starts to receive its innervation from the developing nerves in the area that it starts to develop in. So we said it starts to develop in the region of the cervical vertebrae. That means it's going to receive its innervation from those cervical nerves, specifically the C3, C4, and C5 spinal nerves, which collectively come together to form what is known as the phrenic nerve, which plays an important role in controlling and innervating the diaphragm, so controlling its contractions, and so basically helping with the process of breathing. And so it's kind of important to start to understand how the diaphragm is getting its innervation. Why is it from the phrenic nerve when it sits lower down? And this provides the embryological explanation of that. And your gross anatomy lectures will describe a little bit more its structure and how it comes to be in terms of gross anatomy. So that brings us basically to the end of the embryological development portion of it. And we'll take a quick look at clinical anatomy of what could go wrong if things don't happen exactly as they should during the development of the respiratory system. So right at the beginning, we already looked at, for example, the development of the tracheoesophageal septum. Another thing that's significant here is newborn respiratory distress syndrome. So we spoke about surfactant, remember, and we said the surfactant is produced by the type 2 alveolar cells. It's going to line the alveolar surface, and it's going to play an important role in respiration, acting as the sort of lining really that keeps the lungs open and prevents them from collapsing and falling inwards on themselves when you exhale, right? So in premature births, if a baby is born under or before 35 weeks of development, they are at a risk of developing newborn respiratory distress syndrome. Because what happens is that enough surfactant might not have been produced at this stage to be able to line the surface of the alveoli. So a lot of the smaller alveoli are going to be at risk of collapsing. And if they do collapse, this affects the kind of exchange of gases that will occur, which could lead to newborn respiratory distress syndrome. Now, how this might present clinically in a premature baby is that infant will have very rapid, very labored breathing patterns because, again, they're in respiratory distress. And because the lungs aren't able to fully draw in the oxygen that they need because respiration is not happening effectively, the baby might also present with cyanosis, wherein some parts of the skin, typically around the mouth, around the fingers appears a little bit more blue or bluish in color or purplish in color, because there is this decreased amount of oxygen that's available for the body, in the body, to bind to the hemoglobin of the red blood cells. Now, one way this can be managed is first off is in a mother who's in premature labor, they can be given steroid injections, which could possibly help to improve kind of or speed up rather the production of surfactant, which will in turn help to improve the baby's chances of not going into respiratory distress. Alternatively, the baby itself, if it's born prematurely, artificial surfactant can also be injected in through their airway to reach that alveoli surface to make up for the naturally occurring and existing surfactant, to again prevent this sort of respiratory distress. Next, we also have congenital abnormal hernias. This happens when the pleuroperitoneal membrane does not fully close as the diaphragm is sort of developing. And if there is any sort of insufficiency or issue here that exists in the development of the diaphragm, what could happen is that the contents of the abdominal cavity are going to be able to herniate up into the thoracic cavity. So this is what it would look like in a healthy individual, where the diaphragm would sit here and it would separate the organs that sit in the thoracic cavity from the organs in the abdominal cavity. But if there is a herniation on the left side, or if there is a congenital diaphragm herniation on the right side, all those abdominal organs are able to go up and herniate into the thoracic cavity through the area where that hernia exists. This, of course, ends up kind of encroaching on the space that the lungs and the heart need. It kind of compresses them to a side, or it pushes them, or it displaces them. And because these structures are in the region of the mediastinum, this moving or this displacement or compression of these, the lungs and the heart, this is known as a mediastinal shift, and this can lead to a few issues, right? So, for example, if the lungs are displaced, it could again lead to respiratory distress. Because, it might lead to poor lung development, or even in the development of possibly higher pressure in the vasculature of the lungs, or pulmonary hypertension, simply because the space that the lungs have to develop, that they need to develop adequately, has been compromised. But of course, largely a sort of a diaphragmatic hernia or congenital diaphragmatic hernia can typically be surgically repaired. Okay, so that brings us to the end of the embryology of the respiratory system. I've introduced you to learnanatomy.com before. This is a reference tool that I've created. It covers our gross anatomy subjects. It covers the embryological subjects. It differentiates anatomy by regions or by systems, as we're studying it right now. And I think it's a really handy tool to help to supplement your lecture notes. So I highly recommend that you check that out if you want more information on the topics we're discussing. Now, if you have any questions, feel free to email me directly at antskr@nus.edu.sg. And of course, if you prefer, you can always reach out to me and submit your questions anonymously by clicking on that bit.ly link. Thank you for attending today's lecture, and I'll see you in the next one. Bye, everyone.